Yes. So thank you, Clarissa, for the introduction, and thank you for the organizing committee for the nice conference. Thank you, Clarissa, for putting up the symposium and giving me the opportunity to present the results here. I would like to start by acknowledging, in particular, two of my PhD students, Yardeni and Clara Groot Crego, which made amazing contributions to the results I will present today. And I would also like to express my disappointment to the organizing committee that they chose to remove them from when listing my talk in the program. Anyway, I often start my presentations with such slides, reminding people that the amazing diversity of plant species we see around us, and of course, behind this diversity, we have the process of diversification. But this process does not occur equally across the tree of life. Extreme cases of diversifications are referred to as adaptive radiations. Now, radiations often have an adaptive value when lineages, find and diversify to occupy a broad array of ecological niches in a process which is then adaptive radiation. And examples of such adaptive radiations are well known from animal studies, like cichlid fishes in East African lakes, Darwin finches in Galapagos, or Heliconius butterflies. And in all these studies, the classical theory suggests that actually ecological opportunity is what is really important for these radiations, right? So this almost to be understood as the only required element is this ecological opportunity. And actually, studies from these animal systems in particular, because plant radiations are not so well investigated with, at the genomic level, I think, still. So from such animal examples, we started to understand that the story may not be so simple, that other events or other processes may be behind these radiations as well, as, for example, hybridization, right? We are here for the symposium, or particular demographic events, or genome dynamics, like gene family evolution, and so on and so forth. And in my group at the University of Vienna, we are trying to understand these processes at the, in plant adaptive radiations, and we study in particular three plant adaptive radiations. On one hand, Tillandsia, subgenus Tillandsia, which radiated mainly in Central and North America. We also work on persimmon trees which radiated in New Caledonia to adapt to a wide diversity of soil types, including serpentines and ultramafic soils and so on. And we also work on Nicotiana section saviolentis, which radiated to occupy the arid interior of Australia. But of course, today it is all about monocots, so about Tillandsia subgenus Tillandsia. I will just recapitulate very shortly what yesterday was very nicely shown by Karina's group and including Sandra, that this subgenus originated in the Andes roughly 7 million years ago. And then after at least one long-distance dispersal event, they arrived in North America and roughly 5 million years ago. And there they radiated to produce a high diversity of morphological and ecological traits in a diversity which today includes more or less 300 species in Central America, compared to only roughly 30 species in the ancestral area of South America. Now, of course, the first step in working with any group is to understand very well their phylogenetic relationships and the evolutionary history. And in the case of rapid radiations or adaptive radiations, this may be difficult, right? Because these quick speciation events or quick diversification normally blurs the phylogenetic signal. You have marked incomplete linear sorting around the genome, and very likely a high level of phylogenetic conflict between different regions of the genome. In addition, reproductive isolation often evolves slower than the speciation rate, right? And therefore, gene flow is still possible, and it's often found in such radiations, and this could make the situation even worse. Now, we started to, we wanted to understand better the phylogeny of Tillandsia subgenus Tillandsia, but also for other studies which we are doing, we decided to assemble two reference genomes first for this group. We chose two species which are divergent in their phenotypes, for example, in the chem photosynthesis, and also differ in genome size. So Tillandsia leoboldiana on one side has a slightly larger genome size, 1.1 gigabases, and it's a C3-like plant. And Tillandsia fasciculata is a chem species, and it has a slightly smaller genome size, roughly 800 megabases. So we assembled between 25 and 26 main scaffolds in these reference genomes. These are available from GenBank, and these main scaffolds include the majority of genes present in these genomes. Regarding the genomic landscapes, we observe the expected pattern. Genes are in distal positions on the chromosomes, right? This is shown in this green outer circle here, the gene content. And in the centromeres, you have high content of transposable elements. Of course, the larger genome size also has much more transposable elements, so the genes are pushed even further away with potential implications for their combination rate in Tillandsia leoboldiana. So then we used Tillandsia fasciculata, so one of the reference genomes, and we produced also whole genome resequencing data for 33 species of Tillandsia, subgenus Tillandsia, including some Central American, some South American. Of course, this is a very shallow sampling. We have not even 10% of the species in the subgenus, right? So keep this in mind. And this work was led by Yardeni, and it's currently available in bioarchives and hopefully soon also from systematic biology. Interestingly, the species tree which we reconstructed with Astral, which you can see here, based on roughly 3,700 genomic windows across the genome, does not recover a monophyletic expanded clade K, which was shown with traditional phylogenetics, based on single loci or few loci. But we rather recover two such clades, so two parts of this K clade, K21 here, and K, the other K clades here, so at least two different clades. And I wanted also to point out at the nodes, we have these pie charts which show the level of gene tree discordance. In particular, the red proportion of the pie charts show you the conflicting topologies which are potentially indicating gene flow, right? And we see that these red areas are very prominent in some parts of the tree, but also here at the around the origin of the Central American clade K21, we observed, a high proportion of these topologies indicating the potential presence of hybridization in those periods of time. So we wanted to look more in detail into introgression. So we used the d-statistics and a wide variety of d-statistics or d-statistics-based methods. And you can see here, actually the d-statistics or the main d-statistic calculations shown as a heat map for pairwise species of P2 and P3. These are polarized so that the d-statistic is already positive. You have the colors in this color legend on the x-axis here. You have the d-statistic. So when it's red, it's a higher level of the d-statistic. But on the y-axis, you have the p-value, so the significance, the confidence in these patterns, how confident are we that these are presenting introgression events. And as you see that even at low d-statistic, we can have high confidence that these still represent introgression events. And basically, you have to look for the more stronger colors, and you see that across the radiation, we recover these high levels of introgression. So we can say that this is a pervasive introgression across this radiation. Some of the signals, as you can perhaps notice, are consistent across, for example, between members of K21 clade, which is this additional clade, different than the other K clades, right? So the members of K21 clade and the members of the other K clades, indicating that maybe there is some kind of ancestral nature of this introgression event. And indeed, we used a variation of the d-statistic, which is called F branch, which is showing an excess sharing of alleles, unexpected compared to the species tree, and it will recover like 6.4% of alleles, so excess sharing of alleles. Also explaining that why, you know, these traditional phylogenetic methods perhaps fail to recover the, you know, the two different K clades in this subgenus. We also confirmed this with Phylonet. Again, roughly 6%, very consistent results. Roughly 6% of the genome of this K21 clade seems to originate from the other K clades, where 94% of the nuclear genome originates from the from a South American clade. Apart from the implications for understanding the phylogeography of this of the radiation in the subgenus, we also started to wonder if such you know, hybridization events, introgression events, if they are so pervasive and so you know, both present but also in the ancestry, if they could have played a role in shifts affecting key innovation traits. So we started to look more into the local patterns of introgression along the genomes, and we took examples of species from, well, I will just talk about this example in particular. So Archaeostachys is a Tillandsia from a predominantly C3 clade, but itself it's a CAM species, right? And then we checked if there is any signal of introgression from distant clades, which are actually doing CAM, like you know, represented here by Tillandsia butzi. Indeed, when we look across the genome, we find signals of introgression very punctuated here in chromosome 18. Two regions of high levels of introgression between these two CAM species. And these were, you know, we looked at them in very much detail. We see that the two regions have a low gene content. So, you know, one of the signals is recovered from around the centromere. The other one is, you know, distal in the chromosome, but still in a region of low gene content. This is confirmed also by topology weighting methods, where you see that in these regions, you observed the main topology is this alternative topology, which combines the two CAM species as sister clades. But the rest of the chromosome shows this blue topology as the most persistent, which is the species tree, right? And however, so we started to look which genes are in these regions, right? But we didn't find any particular candidates which are linked to CAM photosynthesis, which is a bit disappointing, right? So we don't know if the signatures of, if this is a signature of genomic landscapes with regions which will have RES recombination around centromeres, for example, will retain signals of introgression for longer times, or maybe this is a signal of selection against introgression in gene-rich regions. So, you know, in the gene-rich regions, you will have less such introgression signals, or maybe if selection for new CAM-related variants, it's indeed required for CAM adaptation, right? So introgression will have a positive effect in trait shifts. So we thought that maybe we don't understand this trait very well in Tillandsia, so we started to, you know, dig deeper in that particular, phenotype. So we went back to our two reference genomes, and we took a comparative genomic approach between Tillandsia fasciculata and Tillandsia leoboldiana. These are some of the, so they're not sister species, but they are quite closely related. They are some of the most divergent CAM phenotypes in the CAMC3 phenotypes in the group, based on, for example, carbon isotopes. And we took, so just as a reminder, CAM and C3, they have a temporal signature, right? With the CAM photosynthesis normally having, so like plants which are doing this kind of photosynthesis, having the stomata closed during the day as an adaptation to drought, or right? And then obtaining carbon dioxide during the night, and fixing it as malate in the vacuole, transporting the vacuole, and then taking from there during the day and using it in the photosynthesis, right? Also, I wanted to remind you that the CAMC3 is not a binary trait. It includes a continuous diversity of phenotypes, you know, going from weak CAM to facultative CAM to whatever CAM. Because of that, we wanted to confirm that these two species are CAMC3. So then we did some metabolomics across time points. So we took 6 time points, we sampled the 2 species, and we confirmed that actually Tillandsia loboldiana, it's doing C3, or in standard conditions is a C3 plant, and Tillandsia fasciculata, it's accumulating malate during the day, and using it, accumulating during the night and using it during the day. So we have a temporal RNA-seq experiment, so sampling across 6 time points across a 24-hour cycle, and we combine this with a comparative genomic approach. We look at, we find always surprising results. This subgenus, as well as the whole family, it's supposed to be a quite stable karyotype with 25 chromosomes, X equals 25, right? But then we sample 2 species, and then we find that one of them has 6 chromosome pairs less. So Tillandsia loboldiana, the C3-like plant, has X equals 19. We also, with our genomes, reference genomes, we recover some genome rearrangements associated with this, contraction of the number of chromosomes. And we wanted to test, and we failed to find any evidence that these rearrangements are linked to any kind of increased adaptive sequence evolution, so DNDS, or any association with any key innovation traits like CAM. However, we did identify that there is a high genome family evolution, especially in Tillandsia fasciculata. So the CAM plant has larger, so it has more multi-copy gene families compared to Tillandsia loboldiana, and these gene families tend to have more members in general in fasciculata. So there is an increase in multi-copy gene members in the species which has actually a smaller genome size, which is counterintuitive, right? So now we did this RNA-seq in a temporal way, and then we find, the expected candidates like circadian clock, circadian rhythm, or genes involved in regulation of ocular transport, malate transmembrane transport, and so on and so forth. This is nice, but what is really interesting is that we started to look at this gene family evolution, and we find that actually the differentially expressed genes recovered are particularly underrepresented in the genes where we have one-to-one orthologs between the 2 species, and they are particularly overrepresented compared to the rest of the genome in the gene families which have more members in the fasciculata, in the CAM species, compared to the loboldiana, and also are overrepresented in these gene families which have an equal amount of members. So they represent an ancient duplication which predates the divergence between the 2 species. So we can conclude that both expanded and ancient multi-copy gene families are actually appear linked to differential expression and also to the phenotype of CAM. Now, Clara took a long effort to and took the entire CAM pathway and mapped on all this on the entire pathway, where we see events of adaptive sequence evolution, differential expression, gene family expansion, or perhaps elevated transposable element insertion rates in introns or around the genes. And it's very complicated to look at, but actually what we see is that we see differential expression in a lot of portions of this pathway, and often this is linked to gene family evolution. So as a summary for this part, we have genomic divergence between CAMC3 species that can be found on many levels, including karyotype rearrangements at the genome level or tree landscape. But actually the differences related to evolution of CAM are mostly found in the transcriptomes, or rates of expression, and gene family evolution. We also were impressed by the small number of changes required. Actually, most of these changes seem to be regulatory in a way, or based on copy number required for such a shift between C3 to CAM. Now, this aspect of gene family evolution related to the radiation or the CAM shifts and so on is not a new idea. We had a paper from the group which was published in 2020 showing that actually in the radiated part of the subgenus you have high level of gene family evolution with insertions or duplications and loss compared to the non-radiated part of the subgenus. So this is, something which was already known. Now, if I have still 1 or 2 minutes, I have some preliminary results from a study trying to link key innovation traits and gene flow. This is done by Clara together with one of our collaborators, Hannes Fardal. They were thinking about a test using genetic data and ecological traits, right, in order to link these 2 patterns. So they calculated trios. So this statistics in trios, including either species which have contrasting ecologies, right? So if gene flow will be between species with contrasting ecologies, the test was polarized so that D is negative. If the gene flow includes species which share the same ecology, this statistic was polarized, so it's positive. So the expectation is that if the gene flow is linked somehow to the ecology, then you would find a lot more positive values of D rather than pairs which will have negative values of D. And indeed, we observed this, we have an increased proportion of positive Ds in this kind of test. And Clara looked into, in particular, into bracts color, flower color, growth habit, photosynthetic metabolism, impounding tank formation, and trichome layer. Now, all ecological traits except for tank formation are positive, are shifted towards the positive values, right? But tank formation and flower color are, however, not significant. And the strongest shifts are shown by trichome layer and growth habit. However, directionality of this relationship still has to be questioned. We are trying to think that if we should bring in geography also to show if sharing an ecology, so living in proximity, provides an opportunity for gene flow, or rather gene flow contributes to the species sharing the same ecology. This is a question which I think it is difficult to answer.
Okay.
So, yes.
Sorry, time's up already. But
good.
We can continue with the questions. Somebody? Yeah.
So, thanks for an amazing talk, great work. Do you think that these ancient hybridization events have an impact on the ploidy levels of the species?
They are all diploids.
They are all diploid?
Yes.
Okay.
Any other questions?
Thanks, Ovidiu. With all these duplication events, particularly the CAM-related ones, do you see that both genes are being expressed in a CAM fashion, what you expect by CAM, or is there some sort of neo-functionalization or sub-functionalization?
So in general, the C3-like plants tended to express just one copy in these traits which are related to CAM, in these genes. But there was some variation also in the CAM plant. Sometimes we saw both copies expressed or several copies expressed. Sometimes it was more than one of the copies was expressed at higher level and the other one was less expressed. So I think it's difficult. I mean, you would need to go even more detail to really understand, and also probably look at the phases of expression, in which time point, and so on and so forth. So there will be, I think, a combination of things.
So thank you for your talk. My question is about the species which lost these 6 chromosomes. Did you say that it was after the hybridization, or is there some
which hybridization?
What was the reason? So
no. So this was not linked. I don't know if this is linked to any hybridization event. Actually, we were very surprised to see it, and we don't have enough sampling to see if this is a deeper or a more recent event. Yeah.
Well, thank you very much again.
Thank you, Ovidiu.